Warahmatullahi wabarakatuh. Good morning and best wishes to us all. I would like to say welcome to Director of Research Center for Bioteknologi, Indonesian Institute of Science, Dr. Puspitalis Dianti, our Honorable guest speaker, Professor Lee Ten from Nanyang Technological University, Singapore, our moderator for today, Dr. Penny Ahmadi, and all participants this morning. First of all, let's be grateful to Allah Subhanahu wa Ta'ala who has given us good health and time so we can attend and participate in the knowledge sharing seminar today. Next, we would like to invite the Director of Research Center for Bioteknologi LIPI to give welcome speech and open this knowledge sharing seminar. For Dr. Puspitalis Dianti, time is yours.
Thank you very much, Mbak Wardah. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody.
Morning.
Good morning, Professor Tan.
Morning.
Yeah, stay healthy and happy. Okay, today we are lucky because we have a special lecture, Professor Tan from NTU Singapore. That would like to share with us about marine cyanobacteria. I'm also microbiologist actually. I work a lot with actinobacteria. Thank you very much for can sharing with us. For the knowledge sharing seminar that organized by Research Center for Biotechnology, LIPI, actually we can discuss deeply. Yeah, we can ask everything. We can discuss, and then the important thing is we would like to collaborate more, with your institute. I think it's a chance to have a collaboration.
For sure.
Thank you, and then let's start for the knowledge sharing seminar. Thank you also for Mbak Penny.
All right.
Should I first open the meeting? So Bismillahirrahmanirrahim. Thank you, Mbak Wardah, and also thank you very much for Ibu Lilis, the Director of Research Center for Biotechnology, for the kind and warm opening for this knowledge sharing seminar, and also for our honorable invited speaker, Professor Tan. Thank you very much for your for being able to give a talk in our knowledge sharing seminar in this editions, July 13, 2021. And in this opportunity, Professor Tan would like to talk about his research. It's titled, may I share my screen? So I will be talking about his background a little bit. So actually, Professor Tan Tong is a senior lecturer in natural sciences and science educations, and his academic qualifications, actually, he graduated from National University of Singapore in 1992, and he got a bachelor degree for honor degree in 1993 from also same university, National University of Singapore, and then he got his master degree from the University of the Ryukyu, Japan. And then after that, he continued his PhD and graduated in 2001 from Oregon State University, USA. And then also the professional qualification of Professor Tan, the latest one, it's at 2011-2015. He was a vice president from the Singapore Institute of Biology. And actually, he has many working experience here, and I couldn't mention a lot because it's too much, but I will mention the latest one, from January 2018 to present, is a senior lecturer in National Institute of Education, NTU. And also he had many honors and awards that he got in the recent one, that in 2018, he got an excellence in teaching commendations. And also in 2019, he got a most cited article in 2019 and marine drugs. It's so excellent. And he has a lot of publications and awards and funding grants from outside and external from and also internal. And also the key areas of his research are marine natural products, marine chemical ecology, and project-based learning. And in addition to that, he also supervised many students from undergraduate student, master student, and PhD student. And it's really many. And if you want to have his paper check, you can go to his website and many paper that is right. Actually, he is not only working on cyanobacteria, but he is also working on actinomycetes and also the invertebrates such as sponge and many marine related organisms. I think that's all for the brief introduction for Professor Tan. And now let's we go to the point for his lecture. So Professor Tan, please, time is yours.
Great. Thank you very much for that kind introduction. So I'm going to share my screen. All right, I hope you can see it. All right, good morning, everyone. Thank you once again, Penny, as well as the Research Center for Biotechnology for the kind invitation to speak to you. All right, so the title of my talk today is Marine Cyanobacteria, a treasure trove of bioactive compounds for drug discovery and development. I'm actually from the National Institute of Education, which is part of NTU. So we are actually a teacher training college. And so basically we train students to become teachers and to feed into the education system in Singapore. And within NIE, the National Institute of Education, we have, academic staff doing research in education research, as well as content research like myself. So I do have a small lab, a small group of people in my lab working on marine natural products. Currently, I have about 2 graduate students, as well as undergraduate students doing project work under me. All right, so that's the background of the National Institute of Education. So for today's talk, because this is a knowledge sharing session, I'd like to sort of introduce to you some background information on the importance of filamentous marine cyanobacteria as a source of drug as well as development. And then I'll talk a little bit about my own research on marine filamentous cyanobacteria. And I'm going to show you just one example of a classical sort of bioassay approach to drug discovery. And perhaps some of you might be quite familiar with this approach. And then I'll talk a little bit about a few projects on the use of innovative mass spectrometric based metabolomic platform in drug discovery, and then some final thoughts on drug discovery from nature. Now, I just want to start off with some introduction, just some general introduction of what cyanobacteria are. I realize that some of you may not be familiar with this group of microbes. Basically, cyanobacteria are classified with bacteria in the kingdom of Menaria due to their prokaryotic nature. And because of their prokaryotic nature, they lack internal organelles, discrete nucleus, as well as histone protein. These are really ancient microorganisms, with oldest known fossil more than 3.5 billion years ago. These are the microbes that essentially puts out the oxygen into the atmosphere. In nature, they occur in small as well as all the way to filamentous form. So it is a filamentous type that I'll be focusing on. All right. Now, in terms of chemistry, it's kind of a genus-based. Cyanobacteria present a genus-based nature of cyanobacterial chemistry, in a sense that on one hand, you have compounds that can pose public health issues, such as microcysteines. These are compounds, cyclic peptides that are produced by freshwater strain of cyanobacteria that can cause liver damage. So it's quite dangerous in that sense that if it gets into our drinking water supply, then you're done for it. So, so the PUB here in Singapore is constantly monitoring our reservoir for possible production of microcysteine from freshwater strain of cyanobacteria. And then from the marine realm, you have really nuisance compounds such as limbia toxin A as well as D-bromoapicetoxin, which can cause skin dermatitis as well as tumor promoter. So these are something that we have to be sort of aware of, especially with climate change. There's increase in algal bloom, microalgal bloom, and some of these bloom can produce such nuisance compound. Now, on the flip side, you have cyanobacteria strain that produce beneficial compounds. So these are primarily filamentous strain of cyanobacteria. They are sources of important pharmaceuticals. And the type of activity they produce ranges from anti-cancer to anti-malarial activity. So this figure here, two pie chart here, shows a breakdown of all compounds produced by cyanobacteria, both freshwater, sorry, it's just marine, from marine cyanobacteria. So I just want to highlight to you that the summary of it is that if you were to look at percentage of marine cyanobacteria natural products by taxonomic order, you find that the filamentous order of Oscillatoralis as well as Nocochilus, these are two main orders that produce majority of the molecule. Now, if you were to look at the percentage of marine cyanobacteria natural products by the generic level, then you will notice that Limbia, all right, there's some revision to its taxonomy actually. Some of these strains are also known as Moria. So Limbia / Moria, as well as certain genus such as your Schizotrix, as well as Symploca, are quite an important source of pharmaceuticals, right? So this is some background on the breakdown of compounds from cyanobacteria. Now, what are the classes of natural products that are found from marine cyanobacteria? Obviously, there are a lot more than just 4 listed here, but these are the 4 main classes or major classes of secondary metabolites or specialized compounds. These are the terms people are using these days, produced by marine cyanobacteria. And I would say that of the 4 main classes of hybrid polyketide, non-ribosomal peptide, you have also the polyketide. Remember the apicetoxin that's belonging to a polyketide structural class. And then, of course, you have the non-ribosomal peptides. And then lastly, where you have the cyanobacteria, also known as the ribosomally synthesized and post-translationally modified peptides. It's a long term. Anyway, so these are the 4 major compounds or classes of compounds. I would say that the majority of the molecule of the 4, the hybrid polyketide non-ribosomal peptide would be the predominant class. Right. So this is just a snapshot of just 4 molecules that is in the forefront of preclinical testing. In terms of activity, they range from, as I mentioned before, anti-cancer. I've highlighted here cyanochrysomate A, which were discovered by colleagues at Scripps, as well as Lagazole, another colleague at the University of Florida. And these 2 are really potent histone deacetylase inhibitors, and they are currently being pursued as anti-cancer drugs, as well as their derivatives. And you have compound here, Tasiamide A, right, which is currently being pursued as a Alzheimer's disease, specifically as a beta secretase 1 inhibitor. And then you have Galelimide A, which is being pursued as anti-malarial compound. Now, you notice that the information given here, I've also provided the genus or the species that the cyanobacteria, that the compounds was isolated from, and they are primarily from Symploca. So this is a picture of the Symploca. I'm not sure you have come across this. This is fairly common in Southeast Asia. I call them Smurf hair because it sort of stands up, and they're as big as my thumb, all right? So they occur seasonally. They don't occur every time of the year. So you need to, sort of capture them or collect them at specific time of the year. But they do produce quite an astounding array of bioactive compounds. Now, in terms of success story, I just had one example shown here, and this is a molecule, dolastatin 10, which was originally isolated by a group in University of Arizona, I believe in the early 80s, called dolastatin 10, all right? And this compound was originally isolated from the sea hare, but eventually was rediscovered in cyanobacteria. So it turns out that the sea hare actually feed on the cyanobacteria diet and sequester dolastatin 10. And this compound, when it was first discovered, was really potent in terms of its cytotoxic activity. And to cut the story short, eventually they generated quite a variety of compounds and synthetic molecule, one of which is known as indoctin, all right, shown here. And eventually this was reconfigured as an antibody drug conjugate, and this is marketed as Acetris for the treatment of Hodgkin lymphoma and anaplastic large cell lymphoma. So this is one of the early success story of a derivative of a marine cyanobacterial compound that's now currently used to treat cancer diseases. And since then, since 2012 or 2011, where this compound was approved, there are at least 3 other synthetic molecule based on dolastatin 10. So this is a list of approved marine derived drugs as of May 2021. So there are about 15 approved marine derived drugs currently in usage. And you will notice that majority of these compounds are used to treat cancer. In fact, in the pipeline, the preclinical testing, there are at least 50 over compounds. So the future of marine drug discovery is bright, all right? So I just want to highlight to you the importance of cyanobacteria as a source of drug by using this table. Okay, so that's it for introduction, and hopefully you have some general idea on the importance of marine cyanobacteria as drug source. I'm going to jump into my research proper now, and I'm going to not go so much in depth onto the research itself, but to give you a breath of what I've been doing in my lab in Singapore. So when I first came back from the US to Singapore, my first question was, do we have any cyanobacteria? That was a very basic fundamental question. In order to start a research program, you need to have your sample. So I went out, and of course, lo and behold, they're everywhere. The issue is, of course, collecting enough biomass. And I just started on culturing work actually. So in the beginning, in the early years, it was just based solely on going out to the ocean, to the intertidal area, and collect cyanobacteria. And because of our water condition, I'm not sure you've been to Singapore, but our water condition, for the lack of a better word, is crap. So most of the cyanobacteria occurs in the intertidal area. And the flip side is good for us, because I don't have to go scuba diving and collect them. I just have to walk out during low tide and collect them. So yes, we do have cyanobacteria in our garden, our backyard, so to speak. And sorry, let me go back to this slide. So I just want to highlight to you the microscopic picture of cyanobacteria. If you notice, Moria producen, all right, these are, I'm not sure if it's clear on your side, you should be able to see stacks of cells like pancakes, right? Stacks of cells stacked up and closed in a sheath. And so these are filamentous cyanobacteria, colonial types, as opposed to unicellular solitary type. All right, so of course, the kind of bioassay that I use in my lab is fairly basic. I use a combination of brine stream toxicity assay, which is a really quick, cheap assay. I think most of the lab in working on in natural product chemistry would probably use this assay to quickly screen their molecule, extracts or fractions for activity. So this is just to show you the range of activity from a concentration of 1,000 to 10 ppm from extracts obtained from different locations. All right, the x-axis, the y-axis. And of course, you would quickly, straight away, realize that this extract, is the most potent in terms of toxicity against brine shrimp larvae. And in addition to this, I've also screened it against the MOP4. This is a leukemic cell line. And again, it was quite consistent with the brine shrimp assay. So that's the key thing about using brine shrimp, is that there's some correlation with cytotoxicity assay. So again, this exact same fraction or extract, I should say, gave the same level of activity compared to brine shrimp. So with that, I proceeded on with getting the compounds out. So this diagram here, or workflow shown here, is a sort of a classical bioassay guided approach of drug discovery, and some of you will be quite familiar with this workflow. And essentially, you start with collection of your sample, and then you bring it to the lab, and then you do an extraction, fractionation using VLC, and then you couple that with bioassay to figure out which fraction to work on, and then you proceed with purification using HPLC, primarily reverse phase. And then you isolate the compound out. And if the compound is known, you're back to square one again, because you can't publish that information. So it's a tedious way of doing things, even though it's people are still using this approach. It is still productive to some extent, but it's a little bit tedious. And of course, the type of bioassay that we use in my lab, and as well as we outsource some of this testing as well, ranges from anti-cancer activity, this is done in my lab, as well as in NUS, anti-malarial, which is at the Pharmaceutical Research Center at A-Star in Singapore, and then antiviral in NUS again. And then of late, I think the past 3 years or so, I started screening my extract for anti-quorum sensing activity. So these are anti-infective activity, you can view it as that, at CELSI, which is a research institute within NTU. So with this approach, I continued with the extraction of that particular extract that was shown to be active. And I just want to show you the first series of compounds called legutamite from Linga majuscula. So yeah, so this is a proton NMR of legutamite A, one of the B major compound that was isolated from a collection of Cyanobacteria at Pulau Hantu, which is just south of the mainland Singapore. And it is it's not too big. It's about 864 molecular weight. From the proton NMR, I was able to deduce that it's a mixture of a peptide as well as polyketide containing moiety. All right. And from 2D NMR, of course, we were able to deduce that it contains 5 standard amino acids as well as 1 hydroxy acid residue. So based on extensive 2D NMR measurements and analysis, we were able to quickly put the structure together. And once you have the planar structure, of course, the next task is to deduce the absolute stereochemistry. And we realize that there are 11 chiral centers. So I use a combination of Murphy's as well as Moshe's method, coupled with the J-base coupling constant values to determine the absolute stereochemistry of this molecule. So you would have noticed too that this molecule is classified. Remember the 4 classes of cyanobacteria? This will be classified as a hybrid polyketide non-ribosomal peptide natural product. You will probably recognize this amino acid sequence here, and then the polyketide sequence below. All right, so this is a hybrid type, which is a predominant class of molecule from marine cyanobacterial compound. Now, in terms of activity, it's not too bad. In terms of the cytotoxic activity, which was tested against 5 cancer cell line. You have the leukemic cell line, You have the A549 is a lung cancer cell line. We have the prostate cancer cell line, the colon cancer cell line, as well as ovarian. The activity is in the nanomolar range. We also tested it against for its anti-malaria activity. It's in the micromolar range, as well as antibiotic activity Pseudomonas against Pseudomonas aeruginosa. So we subsequently isolated legunomide B and C as well. So collectively, these have already been published in the literature. And of course, legunomide A, I would say, is probably the most potent molecule of compared to the other two. And the only difference between legunomide A and B is the absence of a double bond at that position. So it's quite interesting that without the double bond, it's a lot more active. So currently, this legunomide A is used as a structural template to generate synthetic versions of it by several research groups, particularly in China, as anti-cancer, possibly as anti-cancer drug. So that's for legunomide. Now I want to talk a little bit about issues in natural product research that we all face. One of the main issues that we face would be the supply issue. Now, how do we get enough of material for further testing? And if your sample, if you are dependent on nature to provide your sample, maybe that's not the way to go, because some of these cyanobacteria or samples are seasonal. So unless you're lucky in a sense and you go in the right season, you can collect enough of it. But then again, you might decimate the ecosystem. So culturing and genomic approach might be something to explore or to think about. And of course, the other issue is the rediscovery as well as de-replication of known compounds from an extract. So is there a way to quickly, de-replicate the compounds in your extract before starting with fractionation and extraction? And that will cut, that will reduce some time in order to sort of prioritize which extract or which fraction to work on. That will give you a high probability that you end up with a novel compounds, all right? And so metabolomic approach could be a possible way. And that is also an approach that's used for detection of structurally novel as well as related compounds, all right? So that's something that I want to sort of talk a little bit about in terms of the metabolomic approach that I've used in my lab. So there is a sort of shift, a paradigm shift, so to speak, in the approach in natural product research more than 10 years ago. And there's this top-down approach as well as a bottom-up approach in natural product discovery. And in summary, the top-down approach would involve culturing conditions. You can vary culturing condition to elicit the production of new natural products. Of course, you can base your sample with a diverse or unique ecosystem, right? That maybe you can find endemic species that produce interesting molecule. Another way, of course, is to do comparative metabolomic profiling of the extracts, even before doing the tedious isolation of compound. And this is something that I've used in a sort of routine manner in my research called molecular networking. The bottom-up approach is more genomic based, all right? You think of it that way, because you can base on bioinformatic tool. This is something that I've used too. About 3-4 years ago, I started using bioinformatic tool to look at the genome of the bacterial strain, not so much cyanobacteria, but bacterial system, to look for unique biosynthetic gene cluster. And then, of course, heterologous expression of compounds of silent or waking up the silent gene cluster. That's something that you can do too to elicit the production of compound. I don't do that in my lab, but I know there's a research group over at the School of Biological Sciences at NTU that's quite active in this area. And same thing, how you can wake up silent gene in a native host itself. So the bottom-up approach is more of a genomic-based approach for the production of interesting molecule. So I just want to turn my attention to, or rather focus on molecular networking platform that I've been using in my research. Now, what is this molecular networking approach? This chart here shows the MS-based molecular networking workflow, all right? Let me try to explain this to you. So of course, you need to start with your sample, all right? The first thing to do is, of course, you need to obtain an extraction of your sample, all right? And if you are equipped with a high resolution LC-MS system, then that's great. I don't. So I actually outsourced that to a company in Singapore to obtain high resolution MS data, all right, LC-MS data of the extract, all right. So so you obtain a whole bunch of LC-MS spectra, all right, from the extract, and of course you can do some processing of your extract. If you think that the active compound concentration is very low, you can pre-fractionate them to increase the concentration of the fraction so that you can pick up more compounds in that way. So, and then of course you upload the raw file after you obtain the LC-MS file. You upload that on the GNPS, stands for Global Natural Product Social Molecular Networking. It is a supercomputer which is residing at the University of California in San Diego. And basically you upload the files, and within a day, depending on the size of the file, as fast as half an hour to a couple of hours, you will get the result. So basically the supercomputer, I'm not a computer science person, it undergoes a complex algorithm by comparing spectral data or spectra, so to speak. And so similar spectra will be clustered together, all right, in that sense. And the similarity score based on cosine value will be calculated. So a cosine value of 1 means that the pair of compound is identical. While a cosine value of 0.7, for instance, is that the similarity index is only 70%. So that's what it means. And then you can use a software source called Cytoscape to visualize your molecule, right? So the molecule is visualized as node. Each node, each circle, all right, is denoted as a precursor ion. So shown in this example here, you have 2 clusters, all right? One cluster here, and then another cluster here. So basically it's telling you that there are 2 dissimilar clusters of compounds, all right? And if they're clustered together, the compounds are similar or related, so to speak. And of course, you can query the system, right? The GNPS spectral libraries to annotate the node, all right? And you can go crazy with this system because you can overlay the information with extracts from different species or different fraction, and you get quite useful information. And this is something that I've done in another project. I just want to show you how it looks like. So this is how it looks like. This is an overlay molecular networking of close to 1,000 ions obtained from 5 quorum sensing active, inhibitory active marines, bacteria. So this is not cyanobacteria. I just want to show you how it looks like. And at the end of the day, you can query the system and tell you that, okay, what are these clusters, right? That's your first question of dereplication, is that, do we have known compounds in my extract? And so the GNPS will inform you that, yeah, there are some known compounds. And this is a very quick way of figuring out which extract that you want to work on based on the percentage of low hits or high hits if you're looking for known compounds, all right? So, okay, back to the story. Now, so another way of using this molecular networking platform is to quickly detect environmental sample for toxic molecule. Right, that is something that I've used, recently used, and I just want to show you an example using the detection of a toxin from Trichodesmium species. So this is a sample that I originally collected more than 8 years ago at Pulau Seringgat, which is just south of Sentosa Island. It's a tourist spot. And from the lagoon, from this lagoon, we collected cyanobacteria. And of course, we didn't know that it contained a plesiotoxin. I mentioned previously that plesiotoxin caused skin dermatitis. So we collected sample using our bare hand, which was not very smart back then. So, but thankfully, we were safe from the collection. We didn't develop any skin irritation. So we were fortunate enough to find new analogs of a plesiotoxin, and this is just one. The 3-methoxy-dibromo plesiotoxin is one of the 3 new compounds isolated, and we screened them for anti-chikungunya activity. So this is antiviral activity. So this is the first report of a plesiotoxin class of molecule having antiviral activity, which is interesting, because all along we thought of this compound as nuisance compound, that toxin, and they cause dermatitis, tumor promoter, and so forth. So this is the first example that this series of compounds have some useful useful activity. So so this is classified as polyketide natural product. So of the 4, one of the classes is a polyketide structural class, and this belongs to that class. So so I started thinking about, okay, how do we use molecular networking as a technique to quickly screen environmental sample for toxic molecule, especially if they pose public health issues. Then is there a way to inform the public that, hey, better stay away from this area because there's a cyanobacteria bloom, and you might get skin irritant if you get in contact with it. So I started using molecular networking to quickly screen molecule, especially a plesiotoxin compounds present in the extract. So it's a fairly quick method of detection. So this chart here shows you an overlay of 6 VLC derived fraction, and we're able to figure out the plesiotoxin molecular family, right? Clusters of compounds here. And it turns out there are other new compounds, right, that cluster together with dibromo plesiotoxin. So dibromo plesiotoxin is a standard that we see the extract, and we're able to pick out the cluster from it. So it turns out there are quite a number of new compounds yet to be discovered. And of course, we extended this to collection of extract from different locations, right? So this is just to show you the utility of molecular networking as a tool to quickly screen environmental sample for nuisance compound. So this chart here shows the extracts from 5 locations in Singapore. You notice that of late, maybe because of climate change, we've been getting a lot of cyanobacteria blooms that produce toxic compounds. So that's not a good thing, because you might end up with fish kills, especially at the aquaculture area. So that's something that we have to deal with in Singapore. So why are we interested in plesiotoxin related compounds? Of late, there's some interest in this series of compounds as potential drug. And I've just listed 3 examples here. In fact, some of the simplified analog of a plesiotoxin was shown to have anti-cancer activity, which is quite interesting. On one hand, they promote tumor formation, but some of the simplified analog can actually prevent cancer formation. And then you have the possible use of this compound for the treatment of irregular heartbeat, as well as antiviral treatment, right? So molecular networking could perhaps detect, could be used to detect useful compounds or analogs of natural products, of course, coupled with bioassay. That would be a very useful method moving forward, right? So I just want to share with you another story of another class of cyanobacteria, and this is a growing class of cyanobacterial compounds known as the cyanobactin, and this is coming from another species known as Symptoca hypnoides. All right, Now, what are cyanobacteria? Let me just briefly talk about what this class of compounds is all about. As I mentioned, this is a growing class of molecule. It's biosynthesized by the Ribs pathway, and cyanobacteria are typically about 6 to 20 amino acids in length. They can be linear or they can be cyclic, and they are characterized of, further modified by heterocyclization of your cysteine, right, amino acid, as well as your serine or threonine into oxazoline, and further oxidized to the aromatic system such as your thiazole as well as your oxazole ring system. And there's also another modification to cyanobacteria would be the addition of terpenoid moiety. So this would be prenylation, all right? So the prenylation can be on non-aromatic amino acid, such as your threonine, as well as aromatic amino acids, such as your tyrosine, all right? And also N-methylation on histidine, all right? So these are some modification, post-modification of cyanobacteria that will essentially give rise to a diverse range of compounds. And this is produced ribosomally, so this is not non-ribosomal, right? So, so from the extract of Symploca, again, we use brine-shrimp assay, and it turns out that there are two fractions, BLC fractions, that were quite active, tested at 100 ppm, fraction 8 and 9. These are the more polar fraction, eluted with about 100% ethyl acetate, as well as 10% ethyl acetate. Sorry, 10% methanol in ethyl acetate. So these are, fairly active in terms of this activity. And we proceeded with profiling the molecular networking of those fractions. It turns out that there's a cluster of molecules shown here. We call it the trichoramide molecular family of more than 1,000 molecular weight. So this is the first time that I've encountered, in my lab at least, compounds that's more than 1,000 molecular weight. There's no hits in the spectral library of the GMPS library. So we proceeded by isolating a major compound, trichoramide A. So this is the proton NMR, as well as carbon-13 NMR of trichoramide A. And when my PhD student first isolated this molecule, I told her, good luck. This is going to be challenging for you because, it has almost 70 carbon signal. And this is the first time that I encountered such a complex molecule. But she persevered and eventually able to solve the molecule. So, excuse me. So, so together with 2D NMR data, she was able to deduce the regular amino acids, right? Quite a number of amino acids are proline, at least 4 proline units in this model of in trichoramide A itself, and an unusual moiety of cyclotryptophan, all right? So this is a complete structure shown in the slide here. So there is in addition to the regular amino acid, there is an unusual prenyl cyclotryptophan. So this moiety here is biosynthesized from tryptophan as well as prenylinated. Remember, I talked about the prenylation as one of the modification to cyanobacteria molecule. And it turns out that this compound, trichoramide A, has activity against cytotoxic activity against leukemic cell line. This would be primarily against the MOC4 as well as AML2 cell line. And so in addition to proton NMR, she found that using MS fragmentation is quite useful in deciphering the sequence of the amino acid. So this is her thing, she's quite good at deciphering the sequences of the amino acid. So fairly quickly, she was able to sequence, partial sequencing of the amino of the compound itself, and together with HMBC, able to link the sequences up to provide the molecule. And then of course, she finally figured out the unusual moiety, which is consistent with a difference of 254 Dalton. So now, of course, once you have regular amino acid in a molecule, you can use Murphy reagent, right, to deduce the stereochemistry. But for cyclotryptophan, it's a little bit tricky in terms of determining its absolute stereochemistry. So what we did was, this was a little bit tedious in the sense that we first just used regular Murphy's condition in 6 normal HCl, 18 hours hydrolysis of the trichloramide A, and then proceeded with derivatization with a Murphy reagent. We use FDVA, which is a valine amide version of it, but the derivatized tryptophan molecule was not observed through LCMS. So we went back and tweaked a little bit, say instead of 18 hours, we changed it to 4 hours, all right? Because we suspect that the cyclotryptophan, might undergo degradation in prolonged heating. And then we, instead of using the valine amide Murphy reagent, we switch it to alanine amide, so to prevent steric hindrance, so that the derivatization can go forward. So we switched to FDAA instead of FDVA, and we were able to observe the Murphy derivatized tryptophan using LCMS, and that's how we deduce this absolute stereochemistry of the cyclotryptophan unit. And then the mechanism of it is that how does a cyclotryptophan, a prenanated cyclotryptophan unit, get to a Murphy derivatized compound? And during the hydrolysis in acid, basically is a concerted ring opening, all right, of the molecule of the cyclotryptophan unit to a tryptophan, all right, and that will allow derivatization with the Murphy reagent to produce the product so that it can be detected using LCMS. All right, so that's the complete story of the trichloramide series. And of course, she went ahead using molecular networking. As you recall from the molecular network, you saw a cluster of compounds, and she proceeded with isolating other analogs, B, C, D, and E. In fact, D is quite unusual in the sense that it contains a bromine atom that is connected, that is found on the cyclotryptophan unit instead of a prenyl unit. All right, so so this is the last story that I want to share with you. And because of molecular networking, I started thinking about, can we revisit some of the extracts that were previously looked at and which we think that is not interesting? Can we uncover, interesting molecule using molecular networking? And so this is something that we went back to. This collection of Lignin majuscula was collected in 2004, so quite some time back, almost 20 years ago. And when a student of mine did the extraction and purification, he found majusculamide A and B. These are known compounds, all right? It was discovered in the 80s. So immediately he stopped working on the extract and then moved on to another fraction or another extract, so to speak. So this was sort of left on the wayside and not to be thought of at all until molecular networking came along, and we thought, okay, maybe we can revisit some of this extract. So the short of it is that we did, coupled with new bioassay, all right, anti-quorum sensing assay, we're able to figure out the fraction that actually produced the activity. It turns out to be majusculamide A and B containing fraction that were active in the quorum sensing inhibitory assay. Right, this is a proton NMR of a mixture of majusculamide A and B actually. The only difference is in the stereochemistry at this position. All right, so together with molecular networking, before that, so I think that quorum sensing inhibitors from marine cyanobacteria is also something to be explored, because to date, there are maybe less than 10 molecules. Shown here are just 9 compounds that have shown to have quorum sensing inhibitory activity from marine cyanobacteria. So I believe that this is something worth exploring, cyanobacteria as sources of quorum sensing inhibitors. Okay, So basically, we went back to the fraction, and lo and behold, we found the majesculamide molecular family, and they are a cluster of more than 10 or 15 odd compounds. Bear in mind that some of these molecular ions or precursor ions are act up of sodium as well as potassium, so you have to bear that in mind too. But there are other unknown molecule that's related to majesculamide A and B to be discovered. And true enough, my current master student is currently identifying two new majesculamide related compounds. So this is work in progress, but you can see the utility of molecular networking in revisiting some of your old fractions or extracts to look for new compounds. So I just want to end with conclusion is that I hope that my talk has sort of impressed on you on the diverse classes of marine cyanobacterial compound, along with their diverse therapeutic activities. And of course, the use of bioassay in a sort of a classical way is useful. It has been useful and it will be useful moving forward. But I think we have to start using innovative technique approaches in the screening of extracts for interesting molecule, so that you don't spend too much time isolating known compounds. So this is something I just want to leave you with. So in addition to molecular networking, the metabolomic approach, I just started on the genomic approach on cyanobacteria. So I've just started my culturing facility in my lab. And in fact, today is Tuesday. On Thursday, hopefully it doesn't rain, we will go out and collect some cyanobacteria and start some culturing work, small scale culturing, and then using metabolomic approach as well as genomic approach to look for interesting molecule from the collection. And with that, I just want to thank all of you for your kind attention.
Great. Thank you so much for the really impressive research, I think. And right now it's an open session for questions. If you have questions for the audience, you can raise your hand, and then I can ask you to talk directly to the Professor Tan. And while waiting for the questions, it's really interesting that Professor Tan explained us that there is innovative approaches to improve our marine natural product research, especially in cyanobacteria. And also it's really interesting that Professor Tan also used MS-based molecular networking. It's really impressive that it can shorten our time to select the promising fraction that can be used to get a promising compound. And if you have any question, just please don't hesitate to raise your hand, and you can open your mic if you have any questions. And if I have a questions, Professor Tan, it's about the hybrid polyketide and the peptide. It's really interesting because both between peptide and polyketide, maybe they have a different, how can I say, the mechanism in their body or something. Could you explain to me why this cyanobacteria can produce such unique hybrid polyketide and a peptide?
I think that has to do with the biosynthetic capacity of marine cyanobacteria, able to, they contain, unique gene clusters that is a combination of your non-ribosomal polypeptide synthetases as well as polyketides synthesis gene cluster. And so because of the presence of unique type of gene cluster, they're able to produce hybrid, molecule that will really sort of broaden the structural diversity, in terms of combining these two different classes of natural product, polyketide as well as peptide.
So is it possible, I mean, to make it kind of make culture, then we can how induce this culture cyanobacteria to produce such a unique hybrid between polyketide and peptide? Is that possible?
On paper, it sounds feasible, but I'm not sure if anyone has done that. it's going to be quite interesting in terms of sort of heterologous expression of this molecule. And I know of one lab at Scripps that actually did some preliminary work on expression of this hybrid type molecule by teasing out the gene cluster and putting it into an E. coli system, right? Another way of promoting or inducing the production of hybrid molecule would be to use, maybe co-culturing of cyanobacteria, or maybe changing the culturing condition that might elicit some of this silent gene to produce interesting molecule.
Right, great. But I have another problem that when we reculture in our lab, for example, culture bacteria or microbes or something, sometimes they produce a different bioactive after compared to the native. So how we can, maintain their natural bioactive, because this is kind of a tricky, right? Because when we culture different environment, they can produce a different bioactive compounds.
That's something, that is not easy to to sort of figure out. On one hand, if co-culturing produce different set of molecule, is a good thing too, in a sense.
Right.
But if you're looking for specifically a particular class of molecule, how do you sort of maintain that? I don't have the answer to that question, but I think perhaps, I don't know. I mean, heterologous expression might be something to explore, but then you might have to deal with accessory genes and so forth. You need to have some understanding on the genetics, the biosynthetic genetics of this compound before it can even do, getting the gene out and putting it into another whole system for the production of compound. So that's a little bit tricky, and that's something that's in the frontier of research.
Right, thank you. And then for audience, do you have any questions for Professor Tan about his research, or if you have any questions about your research too, can discuss here.
I have a question, Mbak Beni.
Bapak, please.
Thank you. Can I?
Yeah, sure.
Okay. Thank you very much, Mbak Beni. Thank you very much for Professor Tong for nice presentation and very comprehensive. I have 2 or 3 questions. First, maybe we not more chemist, but a taxonomist first, because one question is because about the taxonomies of the cyanobacteria. So mostly right now, they changed the name of the species cyanobacteria, because we compare with like classical taxonomies and the molecular taxonomies, they got a different result. Even the Hilary Paul in Smithsonian, right now they changed mostly his library of cyanobacteria. So in terms of your research, so how do you identify it also for the cyanobacteria using the classical or molecular taxonomies? And the second question about the collection, we know it's very difficult to collect the cyanobacteria because sometimes they attach also with hard coral, and sometimes they attach also with the sediment, and sometimes also they attach together with macroalgae, and we sometimes also make the extraction together with them because it's very difficult to collect the cyanobacteria. So how the technique that you use it, or you just I remember that you mentioned first only only in the beach you collect it, then maybe test they will be attached with macroalgae. So the second part, the third part about the lagunamide, this is very interesting for me because I continue your work with Professor, because we isolated also lagunamide D in Florida during my postdoc in Hendrik Lodge. So I continue also your Lingbia there. And one thing is we got the data is similar cytotoxicity activity. And now we still continue to understand the mechanism of action, the lagunamide D. And I don't know that you still continue about the specific data that can you present to us, or you have the data about also the mechanism of actions for the lagunamide A and C, because I think with Lagunet A and D, we just maybe different only one methyl or one linear structure, I think. So, and that's not make significant activity for cytotoxic activity, even for the HT29 or FY49 and HCT116. So they don't have any significant difference. So maybe that's very interesting also for the mechanism of action, how this macrocyclic deoxypeptide for the mechanism of action for anti-cancer and to propose for the new drugs. So this is my question. Sorry for
No
the three
worries.
question.
All right. Okay. The attempt to answer all your questions. Now, your first question on morphology. So, yeah, from the get-go,
Yes.
when I first started working on marine cyanobacteria, it's always been morphology-based identification. All right. And then, of course, later on, maybe about 10 years ago, and it started using 16S RNA sequences to do the identification. So yeah, so in the beginning, I started using morphology. Then it turns out that everything is linear, based on morphology. And obviously, that's not true because some of the chemistry are quite different. And so when we started using the 16S RNA gene sequences, that provided some insight into the identity or the phylogenetic analysis of some of these cyanobacteria. But because my lab is not a molecular lab, I had to sort of outsource that component, to another lab to help out. So of course, one of my students, she was quite interested, in using a genetic approach to do the identification. So that's something that we intend to do, is that when we collect samples, the first thing we do is that we, of course, collect enough biomass to do the chemistry. And secondly, we collect a small, maybe a thumb-size amount of it, and we store them in RNA later. All right, these are cryopreservative solution so that we can proceed with doing the DNA extraction as well as the phylogenetic analysis of the sample. So that's something that we are trying to do on a routine basis. So yeah, so that's your first question, is that we base on morphology as well as genetic means to do the identification. The second question is on how we collect the sample. So yeah, most of the time the samples, okay, when the samples are occurring as bloom, the issue of contamination with or coexisting with another organism is minimal, even though you do find microalgae as well as marine snails living in the together with the cyanobacteria sample. So we actually do some processing of the sample before we extract them. All right. So we basically do the tedious work of getting rid of macroalgae as well as other organisms that might interfere with the extraction, and try to remove them as much as possible before the extraction. Of course, we're not going to spend the entire day, cleaning the sample. That's way too long. So as long as 80% or so they're clear, okay, that's good enough for us, and then we proceed with the extraction. Your third question is about the mechanism of the leguminate A. That's something that I do not really pursue currently, but I remember sending samples of leguminate A to a molecular lab over at the NUS. This is the Cancer Science Institute over at NUS. And from preliminary studies, it seems to suggest that leguminate A is interfering with the STAT3 pathway. I'm not sure if that means anything to you. It interferes with the STAT3 pathway that brings about apoptosis of the cancer.
Yes.
So something you can look it up, STAT3. All right. So yeah, I'm not an expert on STAT pathway, but you can look it up. So I think it interferes with the STAT3 pathway. Specifically, you know, whether it inhibits any enzymes within that pathway, I don't have the answer to that. But for today, all I know is that it interferes with the STAT3 pathway, and that
Yeah, thank you. Yeah, in further, we
toxins
also using the mechanism of action for leguminoid and institute in NCI, maybe science in US, right? So National Cancer Institute in US, so like the apoptosis pathway. But in our lab, we work also some inhibition for the leguminoid to search the mechanism of action like FIDEC, PNA, something like that. So yeah, that's very interesting to me. Leguminoid A also to compare both with leguminoid B. Yeah.
Let's do this.
Yeah. Thank you very much, Dr.
Thank you. Thanks
Professor.
for the question.
Okay, thank you, Bapak. Is there any questions for Professor Tan that you wanted to ask from audience?
Mbak Penny, boleh Mbak? Saya Dini.
Okay, please Mbak Dini.
Thank you, Professor Tan, for your informative presentation. It's really interesting research. So I want to question, I heard that you will do genomic approach beside the metabolomic approach, if I'm not mistaken. Is that right?
Yes, that's something that I'm trying to start, yeah.
Yeah, so what is your goal doing the genomic approach? So is that you want to screen new compound using a genomic approach, or is that a subsequent research, like you want to know what is the gene cluster encoding your compound or something?
Right.
Okay, maybe that's the first question from me. Yeah, maybe you can answer first.
Okay, thank you. Thanks for the question. That's a good question in the sense that why do we want to use genomic approach? My thinking is that metabolomic approach, of course, is a fairly quick way of screening. You know, sorry, let me backtrack a little bit. Both genomic approach and metabolomic approach could be used as screening approaches, you know, to screen for interesting strains with interesting molecule. But I feel that metabolomic approach is a fairly quicker way of getting into your compounds. Because if you were to screen your strain for interesting gene cluster, it doesn't translate to a molecule. So you know, it just means that, you know, your strain has interesting gene cluster. The question of whether those compounds are expressed in solution or in culture, that's something that you have to test it out using metabolomic approach. So in combination, you know, you could possibly use genomic approach and metabolomic approach to screen it. But genomic approach is not, how should I put it? It's not straightforward in the sense that, you know, you need to have a molecular lab, you know, doing, you know, DNA extraction and so forth. And even before that, you need to culture your sample. You know, you can do genomic extraction using environmental sample, but then you run into problem of, you know, extraction of DNA from other sources. You know, so it's going to be a mixture of genomic material. So a better way, of course, is to culture it. So that's something that I'm trying to do. Culturing of cyanobacteria, again, is not trivial. All right, something that we are trying to figure out based on a paper, also by Scripps. So yeah, so we'll see what happens. So yeah, I feel that if you have to choose, of course, if you can do everything, sure, why not? Genomic approach is great, but it just tells you that there's an interesting gene cluster, you know, and it stops there. But it's good to couple that with some chemical or chemistry technique to quickly assess your molecule of interest, because it all depends on the end goal. What is your end goal? for me, is to look for compounds that could be, that can save life. So if that's the case, then of course you need to couple that with metabolomic approach. Now, having said that, there is a tool, an online platform that can sort of connect or to link genomic approach with the determining of the structure of the molecule based on the gene cluster. All right, that's something I've yet to explore, but that something you can think about is that you can actually probe. If you have the gene cluster, you can actually upload just like GNPS, to tell you the possible structure of that, or the product of that gene cluster. So that's something that scientists can use that tool to look for interesting molecule.
So for you, it's not to screen using genomic approach. If like that, then your plan is not like that, right? Or you will make
yeah. So the way to go is, for me, is because I'm limited my resources, is to use metabolomic approach. Yeah, I mean, for me, it's genomic approach. Yeah, it's interesting, but then unless there's a way to link it up with using bioinformatic tools to to come up with a possible structure, but then it's not 100% hit. So at the same time, you still need to figure out what the actual molecule is, and that could be you could use metabolomic approach for that.
Okay. So based on your experience, so is how long you need to find a new compound using this metabolomic approach until you can, how to say, you can find new drug?
Yeah.
How long you need?
Okay. So for my lab, as soon as because I outsourced my metabolomic what do you call that, platform, by acquiring the LC-MS spectra. I used to use a facility at NTU over at the Lee Kuan Yew Medical School, but they charge like $300 per sample, so it's not cheap. So I started by sending it to an ex-student of mine who's working at Thermo Scientific. So he was kind enough to provide it for free. Why not? So so the turnaround time is, as soon as we send him the sample, within 2 weeks we get the result of the extract on the extract itself. And then once we have the raw data, within a day you can get your molecular networking information. So if you have a lab set up with LC-MS instrument, essentially you can get the result within 2 days. You don't have to outsource.
Okay.
It's that fast.
Okay.
In fact, there's a review paper that the turnaround time can be as short as 3 hours.
So it's like you will always get new compound because you need to check to the GNP database to identify whether it's new or it's not new compound, right?
Right. So but what I just explained is just getting the molecular networking information, right? From that information to an actual new compound, it will probably take another couple of weeks or so, because at every fractionation, you can actually couple that with molecular networking, using that as a screening procedure to track your compounds. Or you can even use just regular LC-MS, that's possible too, to finally track your molecule of interest. But also bear in mind that you see a lot of dots, right, in the diagram. This is a colleague of mine that mentioned to me, some of these dots, he called it ghost dots. They are there, but you can never isolate them. The reason is because the concentration is so low, because of the sensitivity of LC-MS. In order to purify these compounds using regular HPLC, sometimes it's not possible to isolate those dots.
Okay, I think in our institute also, for doing LC-MS, we need to outsource, and it's quite expensive based on my experience. So if I think using LC-MS may be quite expensive. I think the approach, but if you can get new compound in very fast, then it's maybe worth it. Yeah. Okay, my last question.
Yeah.
Okay. Maybe you want to
Oh,
explain
I was going to
another.
say that I think what we all need to think about is, of course, the quality of your sample. I think that's important to think about. Is that, of course, you can go out into nature and collect everything you set your eyes on, but that's not going to be be fruitful, and because you're going to waste resources. So I think from the get-go, the choice of a sample is important. Specifically, what genus or what order of sample that you want to focus on. I think that's important.
So you mean we need to do preliminary screening before we do the LC-MS, right?
Yeah, perhaps you want to look in the literature, as to what, there's any up and coming genus or genera, that provide interesting molecule, any genomic information, based on that, and you can target that in your own sort of ecosystem, to see if there's any those organism in your backyard. I think it's important to have an awareness of, of what you're targeting on. Because in the past, on natural product research, scientists just go out to the field and collect everything in sight, and that's not good, because you are actually destroying or affecting the ecosystem. So I think we need to have a way to collect them, as well as couple that with culturing. I think that's the way to go.
Okay, thank you. One last question. For the analog compound, how is the opportunity to get something new drug from the analog? Is that more promising than we try to find new compound? That's my last question.
All right. So I think for getting new compounds based on the molecular networking approach, and this is something that my PhD student is quite good at, is that based on molecular networking approach, if you have LC-MS data, and if the molecule, for example, cyanobacteria, these are consisting of regular amino acid, you can actually base on MS data to build your structure, to predict your structure, right? And that will lead to generation of new analog, even before going through the tedious process of isolation. So what I'm saying is that MS data, could provide structural information that will lead to figuring out the structure of new analogs.
Okay, thank you very much for your explanation.
Thank you.
Makasih Mbak Penny.
Sama-sama Mbak Dini. Okay, for the next questions. So before go to the next question, please, for the audience, please fill the evaluation form that that has been sent in the chat box. Please go and fill it. And for next question, do you have any questions from the audience? Please, you can ask directly to the Professor Tan.
There's a question in the chat.
Okay, so from Khairul Anam. So do you want to talk directly to Professor Tan, or should we read it for you? Okay. So could you share one or two of your research story problems that you faced during your research and how you solve the problems? I think that's the question.
I guess the problem has to do with I think most of the problems that I've faced with would be a structural problem. Not so much isolation, purification of compound per se, because we use primarily reverse phase HPLC to isolate our compounds out, and to use a combination of RP18 as well as phenylhexyl, and these are columns that you all are familiar with. But the issue that we face with is of a structural type, meaning that the planar structure is fairly straightforward, but when it comes to stereochemistry, sometimes that can be an issue. I've highlighted the story on trichloramide A, where we had to tweak the condition of the hydrolysis and then change to a different Murphy reagent in order to get the product molecule of interest. So and sometimes we need to figure out, for example, any new 2D NMR technique that we can use to solve the molecule. I just attended a Bruker webinar on the use of interesting 2D NMR experiments that I don't use on a routine basis, but could possibly be used to run it on my compound, especially if you want to enhance the intensity of some of this compound that has minute quantity, below 1 milligram. So
Yes.
what do you do besides using Shigemi tube? Are there sensitive experiments that you can use? And the answer is yes, there are. So I think we have to embrace new sort of NMR techniques in order to enhance signals and to get some interesting structural information. And we need to move away or out of our comfort zone because we just use regular HSQC, HMBZ all the time. But there are experiments that are more sensitive that the natural product community might not be aware of, but they are, they are just been, they are what do you call that? The pulse sequences are available, for usage. So so for that, you might want to consider looking at Bruker. I'm not sure your NMR system is Bruker or not, but I'm sure regardless of the brand, you can go to the website and look at some interesting pulse sequences that you can download and to use it on your compound. So yeah, the main issue is structural as well as the small amount that we have of the molecule. Those are the two main issue, and the solution to those issue would be to use new pulse sequences to obtain structural information from there.
Thank you very much, Professor.
Thank you.
No, please. Any other questions from the audience? If you have any questions, please just directly ask. It's great that the Bruker tried to develop such a new technology to help us as a natural product chemist.
Are you a majority of you working on natural product chemistry?
Yes, natural product chemistry, but a little. Now I'm working with a cancer therapy, something like that. So I just want to apply for future. I mean, to apply more the applications of natural product as anti-cancers or therapy or something.
Yeah, that's great.
Yeah. Any other questions? If not from the audience, then maybe should we end up right now? We have time. I think we have several minutes. If you have any questions, please don't hesitate to ask, because this is kind of precious time that you can talk directly to Professor Tan and you can discuss about your problem, I guess. And also, this is something interesting that I have one question, one more question. It's about the cyclic one and the linear one. It's also interesting because when they are cyclized, and maybe the activity also change. But in a cancer itself, that they are easy to recognize the cyclic molecules. But based on your experience, which one is better as the activity between cyclized or linear one, or depend on the?
I've not done any work on that, but from what I've read, is that most of the time the cyclic form is more active in the sense that there's fewer degree of movement or conformational changes, so to speak. Another observation that scientists have noticed is that the higher degree of N-methylation correspond to a higher activity of the molecule. So that's something that medicinal chemists can pursue, is that once you have the natural product, one of the things that they do is to increase N-methylation or to put methyl group on the N nitrogen on the amide bond. So that's something you can do to perhaps improve activity. I've yet to come across paper that sort of when the cyclic compound is in linear fashion, the activity increases. I've yet to come across, maybe they are, but I'm not aware of. But I think that, you know, with my thinking right now is that probably cyclic form is more active. Of course, there are exceptions. You know, you just have to try it out.
Great. So it's about the N-methylation. So it's only N-methyl or any kind of modifications alkyl on the N side or just methyl?
What I've encountered is just methyl.
Oh, really?
For a start. Yeah, of course you can add, you know, more than just one carbon, but for a start you can do that. The other modification that people have tried is increase the oxidation state of the molecule. So for instance, if your molecule has a heterocyclic ring, you know, a thiazolene, you can oxidize that further to a thiazole.
Right.
You know, that possibly could improve activity.
Great. Thank you so much. So I think time is up. So if there's no question, then maybe we can stop our discussion here. And thank you very much for Professor Tan for very interesting, awesome, and very cool research. It's really helpful.
Thank you so much.
Thank you for your time. And also thank you for the audience that joined our meeting today, KSS, and also thank you for our director, Ibu Lilis, for your time and joining our discussion here. And I hope everyone stay healthy and strong during these pandemic situations. Please stay healthy, everyone. And I will give it back to Mbak Wardah. Please, Mbak Wardah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for our moderator, Dr. Penny Ahmadi, and thank you for our guest speaker, Professor Lik Tong Tan, for an outstanding presentation. Next, we would like to invite all participants to join our photo session. For our participants, please verify the videos. Thank you. Please, Mas Naro.
Okay, Bu Wardah, dear Bapak Ibu, to express our gratitude to Prof. Tang for a very great presentations and knowledge to share with us today. Please turn on your camera. I will be having three slides to record, to screenshot. So please be ready. So I will take from the first slide of my screen. Okay, please wait. The second one, please wait. And last screen, I'll be taking the picture for the last screen, and please wait. Okay, I'm done. Please, Bu Wardah, continue.
Thank you. Ladies and gentlemen, we now come the end of our event today. Thank you for Dr. Puspita Lisdianti, Professor Lik Tong Tan, Dr. Penny Ahmadi, and all the participants. See you soon at the knowledge sharing seminar. Thank you very much. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you, Dr. Tang, Prof. Tang.
Thank you all. Thank you all. Stay safe.
Thank you all.
Thank
Yeah.
you.